In this session, we will talk about the protein electrophoresis and different types of immunoglobulins with their structure. Firstly, we will talk about the protein electrophoresis. Let me explain it to you. In an applied electric field, serum proteins separate into fraction consisting of albumin and globulin consisting of alpha-1, alpha-2, beta, and gamma globulin. Immunoglobulin A and immunoglobulin G migrate to gamma globulin fraction. Immunoglobulin M and immunoglobulin D has low electrophoretic mobility. So immunoglobulin D is found mainly in beta globulin, and immunoglobulin M in beta globulin as well as alpha-2 fraction. Now, let's discuss the different types of immunoglobulins. Circulation antibodies are produced and secreted by plasma cell, in bone marrow, in mucosa-associated lymphoid tissue, and in lymph nodes. Immunoglobulin A is predominantly found in saliva, tear fluid, and breast milk. It is also found in secretions from bronchial and urinary tract, where they provide protection against bacteria. Immunoglobulins are expressed on the surface of Mature-B Cell, where they serve as a surface antigen receptor. Some endogenous immunoglobulins are also found on other cells, such as monocyte and macrophages, through Fc receptor. Now, let's discuss the different types of immunoglobulins, with their structures and features. First is immunoglobulin G. Immunoglobulin G comprises the largest portion of serum immunoglobulin. They are divided in 4 subclasses. Immunoglobulin G1, immunoglobulin G2, immunoglobulin G3, and immunoglobulin G4, due to difference in gamma chain. Immunoglobulin G heavy chain has 1 variable and 3 constant regions. Weight of heavy chain is 50 to 70,000 Dalton, having 450 amino acids. Weight of light chain is 23,000 Dalton, having 213 amino acids. Immunoglobulin G3 is heaviest than all other immunoglobulin G subclass due to long series of disulfide bond, which is called as hinge region. So it is good at binding complement. Now, let's discuss the second class of immunoglobulin. Immunoglobulin A occurs as serum monomeric immunoglobulin A, as well as secretory dimer of immunoglobulin A. It means it occurs as monomer and dimer. Dimer means 2 monomer joined together. Immunoglobulin A rarely occur as polymers. Immunoglobulin A dimer are held through the chain called as J chain. Immunoglobulin A has further 2 subclasses, immunoglobulin A1 and immunoglobulin A2. Due to difference in hinge region having disulfide bond, immunoglobulin A are very high in carbohydrates. They don't bind complement. Third class of immunoglobulin is immunoglobulin M, which occurs in pentameric form. Means 5 monomers joined together. 1, 2, 3, 4, and 5. It has molecular weight of 900,000 Dalton. Serum immunoglobulin M occurs as pentameric form. It usually occurs as pentameric form, but rarely occurs as monomeric form. Monomeric form of immunoglobulin M represents the classical surface immunoglobulins on the surface of cell membrane of B cell. Heavy chain of immunoglobulin M have 1 variable and 4 constant domains. 1, 2, 3, and 4. Immunoglobulin M pentamers, like immunoglobulin A dimers, are joined together by the J chains. So immunoglobulin M has high affinity for complement binding. Fourth class of immunoglobulin is IgE. Only a small amount of free immunoglobulin E are detectable in serum, and it binds with basophil and mast cells, and in individuals with allergy to epithelial cells in mucosal lining of bronchial and gastrointestinal tract. Immunoglobulin E plays a role in defense against parasites and in immediate hypersensitivity. Heavy chain of immunoglobulin E has 4 constant and 1 variable region. Last class is immunoglobulin D. So, immunoglobulin D, like immunoglobulin M, is one of the most common membrane immunoglobulin in human B cells, but its function in serum is unknown. This was all about this session. In our next session, we will talk about the concept of alternative splicing.